Well, hello everyone, and welcome to this June edition of our monthly Microtalk series. My name is Gordon Brown, and I'm based here at the MRC Centre for Medical Mycology at the University of Exeter. And my co-chair today is Professor Sarah Gaffin from the University of Pittsburgh. So as always, today we have two fantastic speakers lined up for you. The first is Professor Marie Navarre, who is based at Tulane University in the US. And the second speaker is Professor Zinlin, who's based at Tsinghua University in China. Now, if you haven't joined us before, of course, the purpose of this webinar series is to provide our community with an open access platform in which we can all share in the diversity of the fantastic research that contributes to our field of medical mycology. And every one of these micro talks, we have two talks, each for around 30 minutes. And at the end of that, there will be a joint question and answer session. And can I point out that please place your questions into the question and answer box, which you should see at the bottom of your screen. We won't be using the chat function in this forum. And then at the end of both talks, Sarah and I will tag team and we'll go through the questions. Now, we don't necessarily do them in the order in which they are loaded, because sometimes we pull questions if they're on the similar topic. And they won't show up either on the screens as well, but we will try and get through as many as we can during the time of the question and answer session. And if they don't all get answered, then we forward the questions to the speakers and they can respond to you directly. We also, as you heard at the beginning of this, we also record all talks. So if you or your colleague miss a talk, you can also catch it on our YouTube channel, which you will be able to see on the bottom of the screen. Now, this is also just to remind you, the slide is to remind you that you need to register for the micro talks for every session. That's a procedural operation that works at the center. And so please do that for every session going forward. And just to give you a little bit of a teaser of our up and coming talks in July and August, which you can see on the screen. And finally, just to draw your attention to the Medical Mycology Trainee Series, which is a really great opportunity for trainees and early career researchers to present their work to an international audience. And I also should point out that the organizing committee of this trainee series also selects one speaker each year from the previous people who have spoken, who then present in this forum in our medical micro talks in sometime in the middle of next year. And then finally, just thanks to Jules, Michelle, and Alberta, who run all this in the background and make it all very easy for us as chairs to organize and run this micro talk series. So with the introduction, then it brings me on to introduce to our first speaker. And I'm very pleased to be able to introduce you to Professor Marie Navarre from Tulane University, as I said. So Marie moved to Tulane in 2019 after a long period at Louisiana State University. Marie's research interests in medical mycology really started since her PhD, and her laboratory is focused on investigating the mechanisms of immunomodulation and inflammation by Candida albicans and non-Candida albicans species during host pathogen interactions. Marie's made many key contributions to our field, particularly around mechanisms and impacts of biofilm formation and primarily microbial infections. And this latter part that she'll talk to us about today, which is about how low virulence Candida albicans strains can protect against lethal sepsis. Marie, thank you very much, and over to you.
Great, thank you. Thank you so much, Gordon, and everyone at the Center for Medical Mycology and the University of Exeter for this fantastic opportunity to present to you work that's being done in my lab. The mycotoxins in particular have been super important in keeping the mycology community engaged during this long period of isolation and travel restrictions. I really appreciate this. So yeah, I'm going to be talking to you about work being done in my lab focused on trained immunity and how we're trying to develop that to target lethal sepsis. So first of all, there's two distinct forms of systemic or invasive candidiasis that lead into sepsis. So there's bloodstream infection. This is your traditional intravenous infection model in mice. In humans, this is skin or central venous catheter derived with around 40 to 50% mortality, which is pretty high. It's number 3 in terms of bloodstream infection pathogens in the US. And these infections are predominantly monomicrobial. This is as opposed to intra-abdominal infections. This is the intraperitoneal infection model. In humans, this is GI tract derived either from a perf or an indwelling peritoneal catheter. And there's a wide range of mortality between 40 and 80%. And that often depends on what other pathogens are involved in this infection, because these infections are very often polymicrobial with fungal and bacterial pathogens present. In either situation, this can develop into invasive candidiasis, which then can lead into fungal sepsis. So this is similar to all forms of sepsis. This is a life-threatening organ dysfunction due to an exaggerated or uncontrolled inflammatory response, and the typical biomarkers of cytokine storm, markers of coagulopathy, and multiple organ dysfunction syndrome are present. So we've developed a mirroring model of intra-abdominal infection with Candida albicans and Staph aureus that we've been using to investigate and compare and contrast the different forms of invasive candidiasis. I will point out that the mortality rates are significantly higher in intra-abdominal infections when fungi are involved versus when these infections are bacterial pathogens only. The etiology, Candida, along with many gram-negative and gram-positive pathogens can be present, including Staph aureus, particularly in the neonatal patients. And we and others have observed synergistic lethality with experimental models involving Candida plus Staph aureus, E. coli, Streptococcus, Pseudomonas, Bacteroides, and even MTV. So there's this synergistic lethality we've observed is really broad across many different bacterial groups. So this is a typical survival curve of our infection model. So when we inoculate IP with either Staph aureus alone or Candida albicans alone, we have very limited morbidity and mortality. But when you have the two organisms together, you get this dramatic acute lethality in the mice. So I'm just going to summarize a lot of that work. So by 4 hours post-inoculation, in both the mono and co-infected mice, we see dissemination of both pathogens to both adjacent and non-adjacent sites as far away as the brain. We get massive neutrophil recruitment into the peritoneal cavity, but surprisingly, the local and systemic microbial burdens between these monos that don't die for the most part and their co-infected mice that die in about 2 to 3 days, the microbial burdens are equivalent, which was quite shocking when we were not expecting that. In our co-infected mice only, by 12 hours post-inoculation, we see increases in sepsis scores, hypothermia, the cytokine storm is already in full force. We see significant increases in local, meaning peritoneal, and systemic, meaning serum cytokines, inflammatory cytokines, IL-6, TNF-alpha, and IL-1 beta, as well as the icosanoid PGE2. By 24 hours post-inoculation in our co-infected mice, we are seeing blood chemistry consistent with MODS, which is the multiple organ dysfunction syndrome, as well as markers of red blood cell dysfunction and coagulopathy. Let me just change the pointer to laser. Okay. And then by 48 hours post-inoculation, we're in our co-infected mice only. We see morbidity and mortality with those sepsis scores going pretty high. And in the mono-infected mice, very limited mortality, if any at all. And these infections do eventually resolve. So a major question that arose was whether or not this was unique to Candida albicans, or could other species of Candida that are human pathogens also have exert this synergistic lethality with Staph aureus. So we looked at a range of species, including the close phylogenetic relative Candida dubliniensis that makes all of the various morphotypes of Candida albicans. Similarly, Streptococcus can make hyphae as well. We also looked at Krusei and Paracellosis that can only make yeast and pseudohyphae, as well as Glabrata and as a control Saccharomyces cerevisiae that are relegated to the yeast form. So it didn't really line up with whether these pathogens could form true hyphae. While we did see synergistic lethality with albicans as well as Krusei and Tropicalis, surprisingly, Candida dubliniensis, we never really saw that synergistic lethality. We thought it was an inoculum issue, so we erased the inoculum, and we still only would get 1 or 2 mice to perish from the co-infection. And I won't go into many of the mechanisms. I don't have time. That's not the focus of the talk today. But what we did do is that some of these survivors were left around for about a month. So I just had lab people go back and re-inoculate those mice with the lethal co-infection of Candida albicans and Staph aureus. And to our surprise, we saw a pretty strong level of protection against a very acutely lethal infection. So we were really happy about this, excited, thought we could pursue this as a vaccine, at least to understand mechanisms of protection against intra-abdominal infections. And so we started pursuing those studies with this vaccine model. So our typical vaccine model is we administer the organism 14 days prior to lethal challenge and then let the mice go and then finally euthanize, usually at day 10 to 14 endpoint. We wanted to see whether or not Candida dubliniensis on its own could also induce similar levels of protection, because as you recall, that was Candida plus staph originally in that initial experiment. And we also looked at a number of different Candida species as monoinfections, which the mice can very well tolerate, to see if any of those would also induce protection. And again, we're surprised at these results. So Candida dubliniensis alone was able to induce strong protection, but very different situation with Candida albicans, the homologous pathogen. We only would get about 50% protection. And as you can see, Krusei and Tropicalis fare even worse. And again, these are the same species that are virulent during co-infection with Staph aureus. So they are definitely more damage inducing. I will also point out that dubliniensis has to be live in order for this to function, which will come into play later in the talk. So I'll summarize some of the parameters and limitations of the vaccine efficacy with Candida dubliniensis that we've worked out. First, we can induce cross-protection against the other lethal Candida species during those lethal co-infections, so Krusei and Tropicalis. Other low virulence Candida species can also induce protection. So it's not just dubliniensis. So the Candida albicans, EFG1, CPH1 mutants can also induce protection, as well as Candida auris to some degree, and glabrata, and even to some level Saccharomyces cerevisiae, which is surprising. So this should start giving an indication that we're not dealing with adaptive immunity. Protection lasts up to about 60 days post-vaccination, but wanes after 90 days, another indication that this is not TB cell mediated. And protection is maintained following multiple lethal polymicrobial challenges, so we can challenge several times in a row and still get protection. We've been able to see protection against bloodstream candidiasis, but not vaginal, so the mechanisms involved are very much different between those two infections. And in terms of the vaccination route, either intraperitoneal or intravenous administration works. And I'll show you some of those data. So as I mentioned, and the title of the talk alluded to, that protection is not mediated by adaptive immunity in these animals. So we challenged RAG knockouts, which lack TB cell, and we saw no abrogation of protection when we vaccinated either 14 or 30 days prior to lethal challenge. So that pretty much put the nail in the coffin of going down the road of looking at T and B cell mediated effects. So trained innate immunity, this is non-specific memory immunity mediated by innate cells. It was first discovered in monocytes and macrophages, also in K cells that are trained following exposure to initial microbes or their ligands, followed by epigenetic reprogramming that leads to enhanced responsiveness to a secondary infection. So we went ahead and got rid of the macrophages in this model by administering clodronate liposomes, which will be gobbled up by the phagocytes and effectively kill them. And we were shocked at this result, that with the clodronate depletion, we actually had even better protection than our PBS control. So getting rid of trained macrophages didn't work in this model. So we're still wondering what the cell type was that was involved in protection at this point. What we did know doing flow cytometry of the peritoneal cells in naive and vaccinated animals, we looked at both neutrophil and macrophage populations in that peritoneal space, and we found there were twice as many or double the amount of Ly6G positive neutrophils at the time 0 point. So this is 14 days following immunization. So 14 days later, there's still more PMNs in those animals. And this rises dramatically following lethal challenge in the protected mice. Considering this is a model of sepsis with dramatic inflammation, this was kind of surprising to see this many more neutrophils when we're seeing, basically reductions in that cytokine storm in these vaccinated animals. But we thought perhaps these GR1 positive, Ly6G positive cells were actually involved in mediating protection. So we used GR1 depletion, and finally we were able to get a dramatic reduction in protection versus the non-depleted animals or isotype control treated animals. So the GR1 positive PMNs have never been previously shown to play a role in trained innate immunity. This was very novel for the field, and we were wondering, well, is this just unique to our polymicrobial intra-abdominal infection model, or is this across the board with other types of Candida infections, that this cell type is the predominant cell type involved in trained innate immunity? So as I alluded to, we can administer this vaccine either IV or IP, and then the two different models that we have used are the intra-abdominal infection model and then the intravenous bloodstream infection model. So these are the results that I just showed you with IP immunization in our intra-abdominal infection model and GR1 cell depletion. This is what our sepsis scoring typically looks like in our naive versus our immunized animals. This is with intravenous immunization, where we're comparing intra-abdominal versus bloodstream infection model. And this is just a mono Candida bloodstream infection model, not a co-infection model. This is your stereotypical IV Candida feet up, feet down model. And as you can see, this intravenous immunization resulted in very strong protection against both of these types of infections and correlated nicely with what we see for the suppression and the sepsis scoring. I'll also point out, as I just mentioned, that we see reductions in inflammatory cytokines, not increases. So this is again suggesting these are both models of fungal sepsis, and that to get protection in the model, perhaps that you need to have reductions in the cytokines. So we went ahead and then depleted both macrophages and GR1 positive cells from these two types of infections. And we found that with clodronate depletion, we really don't see any significant reduction in the protection elicited by intravenous administration of the vaccine. But we see a dramatic abrogation of protection when we administer that GR1 antibody. So in either case of fungal sepsis, whether it's bloodstream infection or intra-abdominal infection, it's really the GR1 positive cells that are mediating trained innate immunity in our model. So how do these GR1 positive, what we consider to be neutrophils, mediate trained immunity against fungal sepsis when their lifespan, as we know, is pretty short, between 24 and 72 hours? And we're getting protection up to 60 days. So we wondered whether the GR1 positive cells were actually myeloid-derived suppressor cells. These are immature cells of a myeloid origin, and they're a heterogeneous population with a common biological activity, which is immunosuppression. And there's two different varieties, the G MDSCs and the M MDSCs. So these are phenotypically similar to neutrophils and monocytes, very hard to distinguish in the murine model. They are characterized by co-expression of GR1 and CD11B, and importantly, they have longer lifespans than either inflammatory neutrophils or inflammatory monocytes. I just want to give a little primer on the GR1 marker that we have been using to deplete cells. This will actually pick up both G MDSCs and MDSCs. GR1 is just a murine antigen, GPI linked marker. It's the antibody against GR1 recognizes two distinct molecules, Ly6C and Ly6G. So the monocytic MDSCs are Ly6C positive, Ly6G negative, and the granulocytic or neutrophil-like MDSCs are Ly6C negative, Ly6G positive. So by using the GR1 antibody, we're really targeting both potential types of MDSCs. So we then used either Ly6G or Ly6C depletion in these studies, and we saw a significant abrogation of protection when we administered the Ly6G antibody, similar to our pan-GR1 depletion, whereas Ly6C did have some effect, but it was not statistically significant. So this is really pointing to more of the importance for these granulocytic MDSCs in mediating the trained innate protection. So we went ahead and looked at all of the different permutations of the models that I've just been showing you, and followed that up with just Ly6G depletion to really show that it was a role for the granulocytic MDSCs in mediating protection. So I know this is a busy slide. We have the isotype control in the open circles and Ly6G depleted in the closed circles. And if your eyes scan through here, you see these are all the isotype controls and the various permutations of intravenous vaccination, intraperitoneal vaccination against BSI or IAI. And in all situations, we have a dramatic reduction in protection with our Ly6G depletion. We also did flow cytometry to show that the GR1 and Ly6G depletions actually targeted the granulocytic MDSC populations rather than the monocytic MDSC populations in both situations. So this really strongly supports this being mediated by the granulocytic MDSCs. So how does Candida dubliniensis or any of our other low virulence Candida species actually train these MDSCs? We know that development of mature MDSCs is a two-step process, starting in the bone expansion of these precursors in the bone marrow, followed by activation steps, which induces emergency myelopoiesis, calling out these MDSCs into the periphery. And they have various mediators that they produce in order to mediate the end phenotype, which for the cancer field is T cell suppression. Obviously, we're interested in some other mechanisms that I'll get into in a little bit. So the first question, does Candida doubliniensis actually access the bone marrow to interfere or participate or induce expansion of these MDSC precursors? And we've never been able to find any data showing that Candida albicans can even access the bone marrow in a murine model. So we just plated out the femurs of these immunized animals at 24 and 48 hours post-immunization, and we were surprised to see very high levels of Candida doubliniensis in the bone marrow, up to 10 to the 3 per mil of bone marrow content. And this was sustained even at 48 hours post-inoculation. So as I mentioned, there's a number of different low virulence Candida species that can induce this protection, including this Candida albicans double mutant that is unable to form hyphae, along with Oris, glabrata, and like I said, to some degree, Saccharomyces cerevisiae, which is much greater than Candida albicans. So we decided to look at the CFU penetration of all of these different vaccine strains, and we found that Candida doubliniensis had the highest CFUs compared to all of these other species that we saw were able to induce some level of protection, followed by the EFG1 CPH1 mutant. So we decided to plot out the percent protection, which is shown in the parentheses here, against the CFU at 48 hours. And you can see there's a really, really strong correlation between the ability of these strains to induce protection and whether they persist in the bone marrow at 48 hours post-immunization. We've also been asked repeatedly, how long does Candida doubliniensis actually persist in that bone marrow? And we see that it is gone by day 7 post-immunization, and we routinely challenge at day 14. So it is very much out of the bone marrow by the time we administer a lethal challenge. So we also noted after doing some bone marrow histology that protection was also associated with increased bone marrow cell proliferation. So this is the bone of a naive, uninfected animal. And this is what happens after you administer lethal challenge. By 24 hours, you can see there's a disruption in some of that cellular architecture within the bone marrow, as well as what appears to be some level of phagocytosis. This is very much in contrast to what we see in immunized bone marrow. So these mice have been immunized 14 days prior with Candida doubliniensis, and you can see that the bone marrow is just chock-a-block full of cells at this time point. Even more so than when we immunize with the less protective Candida albicans strain, DAY, which is a variant of SC. If I magnify this to a higher level, you can see that most of these cells in these immunized animals do have a granulocytic phenotype. To me, these look like very immature neutrophils, the banded neutrophils that you sometimes see coming into infections very early on. So to look at this in a bit more elegant and specific way, we started doing flow cytometry of the hematopoietic and stem and progenitor cells in the bone marrow following immunization. And all of this elegant work was done by a postdoc in my lab, Shannon Escher-Riggi. So we started looking at the cells in this purple box here, which are the HSPCs. These include the long-term and short-term stem cells, as well as multipotent progenitor cells, which can be further divided into myeloid biased and lymphoid biased progenitor cells. So these are termed LKS cells. First, I will point out that there is a lower limit of inocula that we can administer in terms of our immunization with Candida doubliniensis, at which point we lose protection. So if we go below around 10 to the 6, we lose ability to protect. But what we did notice was that this expansion of these LKS cells tracked very nicely with our inocula that we use for Candida doubliniensis and the ability to protect. So that expansion occurs with rates or inocula that are protective. We then depleted the HSPCs using an anti-c-KIT antibody, and were able to abrogate protection. So we very much need these bone marrow precursor cells in order to have protection in this model. The other thing that we started looking at following our lethal challenge is whether or not we see Ly6G positive GMDSCs expanded in that bone marrow. So this is the activation step. So we looked at total GR1 positive cells in our naive and immunized animals, and by 24 hours post-lethal challenge, we see significantly more GR1 positive cells in the bone marrow. We then looked specifically at the G versus the M MDSC using Ly6C versus Ly6G, and you can see that we get an expansion of the G MDSCs at the expense of the M MDSCs in the bone marrow, once again supporting what we've seen with the depletion studies. So we know from the cancer literature the different types of mediators that are produced by MDSCs that mediate effector function. So we wanted to see if we could detect any of these effectors in animals following lethal challenge or following elicitation into the peritoneal cavity, and did RT-PCR. And we can see that following immunization, we see significant increases in the ARG1 marker, as well as reactive oxygen species, genes involved in reactive oxygen species. So again, this points to them being more of a GMDSC phenotype. So the gold standard functional assay to show that your cell is an MDSC rather than an inflammatory neutrophil or an inflammatory monocyte is to see whether or not they can actually suppress T cell proliferation, which is kind of ironic since our model doesn't involve T cells, but this is the functional assay we needed to be able to demonstrate that these cells are in fact MDSCs. So we isolated GR1 positive cells from the peritoneal cavity of immunized mice by elicitation with Simusan, and then co-cultured those along with splenocytes. So this is when you have no GR1 positive cells in that culture, and the T cells go from, this is prior to stimulation, and then upon stimulation, they all proliferate. However, when we add GR1 positive cells to the T cells, you can see that that inhibits that proliferation to some degree. So we can plot that as a proliferation index. You can see that the proliferation index is lower from GR1 positive cells from immunized mice rather than naive mice. And then that can be calculated as a % suppression. So we can say with certainty that our GR1 positive cells in our immunized mice that are called into that peritoneal cavity are in fact MDSC effector cells. So there has been some work done from other groups looking at the interaction of fungal pathogens with MDSCs, and it is known from those studies that Candida can be recognized by Dectin-1 and then signal through Card9. So we decided to see which of the fungal PRRs are involved in mediating induction of these protective MDSCs. So we inoculated wild type Dectin-1 knockout, MIghty88 knockout, and Card9 knockout animals, either naive or unvaccinated. I'll point out that these are B6 backgrounds, so we had to give a slightly lower inocula of our intra-abdominal challenge, so they don't die quite as quickly in this with this lower inocula. But what we did find was that we only saw significant abrogation of protection in the Card9 knockout. So this indicates that it's probably involving the C-type lectins, but it's multiple C-type lectins. So if we only knock out Dectin-1, that does not have much of an effect on abrogating protection. So I've mentioned these effector functions several times being important for T cell suppression, but our model is a sepsis model, and we're not concerned with what's happening with the T cells at these early stages. So what we really were interested in is anti-inflammatory cytokine production that is known to be produced by MDSCs and how that can be involved in suppressing sepsis. So we did kinetic studies looking at peritoneal lavage samples for IL-10 levels following lethal challenge. And then you can see in the vaccinated mice, we have significantly higher levels of IL-10, which we were expecting since this is a very strong anti-inflammatory cytokine required for suppressing sepsis. And then our studies in IL-10 knockout mice really backed this up. So we saw an abrogation of protection in the IL-10 knockouts compared with the wild type mice. So this cytokine is extremely important in mediating this protection. And this tracks very nicely with what we're seeing with sepsis scoring, where our IL-10 vaccinated animals have very high sepsis scoring as well. So another key piece of evidence that we needed to show that these MDSCs were actually the cells mediating protection in these studies is to be able to transfer that protection cellularly. And so the easiest way to do this, and the way I've done it in the past, is just through adoptive transfer. You isolate the cells and you adoptively transfer them and show that they have they can confer the protection. So we've done this so many different times over the past 5 or 6 years. We could just write a paper of negative data. So we've tried adoptive transfer of cells from different sources. These have all been immunized mice, immunized with various strains, various doses. We've tried to administer the cells IP, IV, but they've always been unactivated, either total cells from the bone marrow or spleen, or GR1 positive isolated or Ly6G isolated cells from these tissues. And they've just been, one time we'll get results, and the next time it just won't repeat. So we wondered, and by reading some of the literature in the BCG field, whether or not it wasn't just the MDSCs in the bone marrow that were helping to mediate protection, but also perhaps the stromal cells, since we know Candida dubliniensis accesses the bone marrow and could potentially influence those cells as well. So to investigate that, we had to do a bone marrow transplant experimental design, which will assess hematopoietic versus stromal cell involvement. So we have a number of different groups here. It's a long time course to allow for irradiation and reconstitution. I'll show you that the first control animal, this is naive into naive, and then we administer a very short vaccination, that 2-week that we normally use, that is really strongly protective. We expected high survival in these mice. We had mice that we administered the vaccine way in the beginning, along with all the other donor and recipient groups. So they got CD-vax, but they got no bone marrow transplant, but they were allowed to sit for 12 weeks. We were expecting maybe moderate protection because that's a 12-week, and we know that the protection goes away around 90 days. We then had CD-vax donor into CD-vax recipient. Again, this will be a long vax, hoping to see moderate protection, naive into naive, low protection, CD into naive, low, naive into CD, if our idea is correct that the stromal compartment is required. And fortunately, that is pretty much what we saw. So this is our number 1 group. We saw very high protection when we administered the vaccine 2 weeks prior, which we should expect. And that also shows that our bone marrow transplant was successful. The moderate group was our CD bone marrow into CD recipient mice. So that's this group, moderate. Strangely, the 12-week vax with no bone marrow transplant, similar to what we saw before, was not that great at protection. So that's when we lose protection typically, but very weak protection in all of these other groups, naive into naive, CD into naive, naive into CD. So this really answers the question of why the adoptive transfers had not been successful. So based on all this work, we have a novel model for innate training called trained tolerogenic immunity. In this situation, this is mediated by GR1 positive cells as MDSC. It requires IL-10 and limits inflammation and sepsis. So the initial stimulus is a low virulence Candida species that we can consider a live attenuated vaccine. And then our second stimulus is sepsis. In investigating the translational potential of trained tolerogenic immunity, we started looking at whether fungal beta-glucan could also induce this, because we've been using a live strain. And this is all work done by Amanda Harriet in my lab. She's a PhD student, and she was able to show that both beta-glucan and zymazan were able to induce very strong protection in the intra-abdominal infection model, which was fantastic. This took a lot of effort on her part. Is the protection induced by beta-glucan mediated by the GR1 positive cells? We did the collagenate or GR1 depletion. And once again, we are getting abrogation of protection with GR1 antibody depletion in both zymazan and beta-glucan, with a moderate role for macrophages with beta-glucan immunization. Can TTI be extended to other CLR ligands? Yes, beta-glucan compounds are effective. Can the fungal TTI, again, can be protective against other infections associated with pathological inflammation? So we did a sterile sepsis model as well as a different type of intra-abdominal infection model. I don't have time to go into the pulmonary infection work that we've been doing at this point. So our traditional sterile sepsis model, this is just high dose zymazan. If you administer 1,000 mg IP to an animal, it will induce acute septic shock. And we were able to induce protection by immunizing ahead with Candida dubliniensis, and that protection was once again reduced with the anti-GR1 depletion, and really no effect of adding collagenate in. And this tracks really nicely with the sepsis scores. So another protection against another type of sepsis. So it requires GR1 cells, not really macrophages once again. We also developed another polymicrobial infection model with a gram-negative pathogen E. coli. And once again, Candida dubliniensis immunization induces very strong protection. And this protection is again mediated by the GR1 positive cells. So to finish up, we are also interested in understanding whether other live attenuated vaccines can induce trained immunity or trained tolerogenic immunity as a non-specific benefit. There were some clinical trials for BCG and MMR against COVID prior to the advent of the COVID-19 vaccine. But can TTI be a clinical means to mitigate against sepsis? We know that this isn't a lifelong immunity, but could live attenuated vaccines be considered a systemic probiotic? And can we use abiotic inducers as novel vaccine designs by including beta-glucan in those preparations? And I'd just like to acknowledge my laboratory and my collaborator, longtime collaborator and husband, Paul Fidel, our former lab members who did a lot of this work, as well as collaborators and colleagues at Tulane and UT Memphis and University of Michigan, and all the people in my lab that made this happen.
Thank you, Mari. Wonderful talk. So to the audience, as a reminder, you can put questions for Dr. Navarre in the Q&A, not the chat. We were not monitoring that. And please keep the questions coming. We will handle them all at the end, excuse me, in a group discussion, back and forth discussion with our second speaker, Dr. Singh Lin. So it is my great pleasure to introduce my old friend who I just calculated I've known for half my life, Singh Lin, coming to us from Tsinghua University in China. He did his undergraduate training in China and then came to the US to do a PhD at the University of Texas in Houston. And our paths crossed when he moved to San Francisco to the UCSF Gladstone Institute to do postdoctoral work with Werner Green on T-cell receptor signaling. He did really outstanding work there. And our paths then recrossed about a year later when he joined the faculty at the University of Buffalo, part of the State University of New York, in their microbiology department. And I had preceded him there. I was in the dental school and he came to the medical school, and we started having joint lab meetings. And Singh decided, and this was in around 2000, when we thought we knew everything already about the T-cell receptor signaling pathway. Singh said, no, there's a lot more to know. And I thought he was crazy and told him so. And in remarkably short order, Singh's new lab at University of Buffalo discovered a hitherto completely unrecognized pathway in the T-cell receptor, namely the involvement of a protein that originally was called KARMA1, I believe is now CART11, which then binds to MALT1, BCL2, 3, whichever it is, the CBM complex. And everyone in the fungal world knows this, of course, as being downstream of dectins. But it was Singh who then, having discovered KARMA1, went on to start to look at other members of the family. And in one of the most landmark papers, I think, in our field, certainly one of the most important mice that we have, was he found that CARD9, not necessarily important T-cell receptor signaling, but absolutely critical for antifungal signaling. And so that really catapulted him into our fungal world. He has really just been an outstanding scientist in the T-cell fungal physiological world. He, after Buffalo, he moved to the MD Anderson Cancer Center, did some really stunningly terrific work there, and about 8 years ago now, relocated to Tsinghua University in China, where he is, I forget his title, it's very long and impressive. So I won't take him any more time. Singh, please take things away. And to everyone listening, if you're just joining us, put your questions in the Q&A. We will try to get to as many as we can and have a great discussion at the end.
Okay, great. Thank you very much for Sarah for your introduction. So I'm going to show my screen and then about my talk. So my talk today is about fungal infection. And so as Sarah mentioned, and my lab is a signal detection, signaling lab. And during last more than 20 years, and then we keep doing different signaling work. And actually a small part of my lab has been interested in the fungal infection induced immune response and how the different signaling component and pathway in this area. So, and after I moved back to China, and then although most of my labs right now is working in these T-cell therapeutic and this anti-tumor work, but I still maintain a small group working on the fungal immunity. So today's work is title is about LTA4H and LTB4 and BRT1 axis played a critical role in antifungal immune infections through regulating neutrophil function. Okay, so and as we know, these invasive fungal infection are common cause for mortality and then morbidity in the high-risk patient, and then those fungi, including the Aspergilla and then Candida albicans, and then also Cryptococcus and etc. So those are major people risk for this is with a critical illness and then immunosuppressive disease and a broader spectrum of using the broad spectrum of the antibiotics or some surgery. So this story was come from, and I had one of my friend and also collaborator in Shanghai Fudan University, and he's a physician, Dr. Li Pingzhu, and he's a physician. And then he found in his a cohort of patient with these non-HIV per patient with a Cryptococcus infection. They have an immune reconstitution inflammatory syndrome for this patient, and they found several SNP. And actually, found in the LTA4H, these genes, non-coding region probably regulate the expression. And so with that, and then he talked with me, say whether we can do something together. And then so we decided to look at that. So LTA4H actually is an enzyme that synthesize leukotriene B4, LTB4, so that in response to variety of stimulant, and then effect is a high affinity through the receptor BRT1. So these enzyme control the from LTA4 and then change to the LTB4. Okay. So then this LTB4 will work on the trigger these let me use the pointer here. Sorry, laser pointer. Okay, so this LTB4 will work on the BRT1 to trigger the signaling, so that play the very important role. So that this LTA4H is a cytosolic protein and widely expressed in almost all the mammalian cell. But the sources of this LTB4 is made in the neutrophil monocyte and then other myeloid cell. So this LTA4H actually has two bifunction. One is expo hydrolase activity, and then the other one is amino peptide activity. So previous study and suggest these enzyme actually played an important role for regulating this. One part is played an anti-inflammatory role, and then another activity is played the role for the pro-inflammatory. So it's a balance for these two functions. Okay. So the connection for this enzyme to the fungal infection has been shown previously in the one work from this Oliver Wade's lab. They found this Aspergilla fumigata can generate a toxin, gliotoxin, that is a suppression of the LTA4H. So therefore, regulate the antifungal immune response. So that has been suggested, but the detailed mechanism is not being well studied. So as a signaling person, so we decided to look at these things. Then so we generated a knockout for LTA4H knockout. So as the typical trick where the way we do this is we take the knockout mice and then challenge with siabecan and then look at the survival. So as you can see that with the LTA4H knockout, now these mice is highly susceptible to siabecan infection. Okay, and the fungal load in the kidney is very high. And then if you look at these fungi loading in kidney, so you can see a lot of fungi in the kidney. So it was a damage. Okay. So then to figure out what cell type probably played an important role. So okay, we also see the high level of inflammation detected in kidney, and then with these knockout mice after fungal infection. And as you can see that in these percentage of the neutrophil is greatly increased in the kidney, and then also there's a lot of pro-inflammation cytokine like IL-6 and then IL-1 beta in the kidney. There's an increase, and then with this, and then you can see a lot of these fungi loaded in the kidney. Okay, so then, because the LTA4H is majorly expressed in the neutrophil and or monocyte or macrophage or dendritic cell, so we want to know what cell type played an important role. So we also generate the LTA4H flox mice. So we did a conditioned knockout and then flox them out in the CD11C Cre and or lysozyme Cre mice. So that in this case, you see there's no difference. So neither these DC or macrophage probably play important role in this knockout. However, when we cross the mice to the flox mice to the S100A10 Cre mice, which is neutrophil specific Cre, now we can see these mice is highly susceptible to the siabecan infection, to systemic siabecan infection. So these data suggest that the LTA4H played an important role in the neutrophil. And so to summarize this part, so we found that LTA4H deficient mice were more susceptible to systemic siabecan infection, and then these deficient mice exhibit a high level of inflammation after fungal infection. And then we found that the role of this LTA4H is majorly in the neutrophil because we use the conditioned knockout to show this function. So with that, and the next question is, and then how is what's the mechanism? So, and as you can see, wild type and knockout mice, then if after infection, the neutrophil level is increased. So suggests that although there's a lot of neutrophil in the kidney, but somehow the function has a defect because they cannot clean up the fungi. So you can see a lot of fungi load in the kidney. Okay. So this suggests that maybe somehow this pathway LTA4H played some role in the regulation of neutrophil. So we want to know whether that's an intrinsic role in the neutrophil. So when we look at that LTA4H deficiency and then see whether they affect the beta-lactin receptors, so that's the first thing we asked. So we found that actually all these lectin 1, 2, 3, there's no significant difference. So that LTA4H deficiency does not significantly influence the expression of pattern recognition receptor and with or without fungal infection. So, and also TOR, that's not the huge difference. So, but somehow the fungal infection is defective. So, and also this LTA4H deficiency only slightly affected some chemokine such as CXCL2 or some CCL5. So that's that's something we found. Okay. But when we look at this swarming effect for the neutrophil swarming effect, so this green fluorescent, this is a green fluorescent C. albicans. So we infect the mice and then look at the kidney section. So you can see this heterozygote mice, and these C. albicans were infected in the kidney, and also LTA4H knockout. Now you see this section. There's these neutrophil and swarming to this effect. So you can see this neutrophil swarming effect. But in the knockout, now you can see swarming effect is dramatically reduced. And also, the other thing is that if we look at the ROS production, and as you can see, this red is a knockout, then blue is a heterozygote. So you can see, and with the heat kill the C. albicans to stimulate the neutrophil. Now we can see actually the knockout cell neutrophil, the ROS production is significantly reduced. So most striking thing is LTA4H deficiency and reduce the phagocytosis activity for the fungi and also fungi killing. So as you can see, this is heterozygous neutrophil, and then there's the knockout neutrophil. And if we use green fluorescent C. albicans and then measure the phagocytosis, and as you can see, this phagocytosis is dramatically reduced, and with 15, 30, 60 milli. So you can see this phagocytic effect is greatly reduced. And also C. albicans killing efficiency also reduced in the knockout mice, knockout neutrophil. However, these things that this deficiency for the phagocytosis is not a general phagocytosis effect, because if you use APC coded beads and then these latest beads, and then we don't see the difference for the neutrophil phagocytosis effect. So that means it's a fungi specific phagocytosis. So as I mentioned, LTA4H and then will generate LTB4 that would activate the pathway to induce the receptor BLT1, then will activate downstream signaling such as ERK activation or the MAP kinase activation. So when we look at this neutrophil and from LTA4H heterozygote or knockout mice, and what we found is these ERK activation and is a very significant reduced and also partially reduced for P38 and then JNK activation. So that suggests this pathway somehow is defective. So, and then surprise to us is the macrophage from LTA4H knockout. There's no any defect for this after with the fungal infection. So with a different time of fungal challenge for the macrophage, then you don't see any difference for the ERK activation and also other. So this suggests whatever we see here is very specific for neutrophil. Similarly, if you look at the phagocytosis effect, either use a bone marrow-derived macrophage or peritoneal macrophage, there's no any difference for the phagocytosis effect for this macrophage. So whatever we found, this LTA4H activity, it's only in the neutrophil. So because this LTA4H and then will generate the LTB4, so therefore we would use this LTB4 to treat the pre-treated neutrophil. So as you can see in a non-treated neutrophil, that this phagocytosis effect is huge difference with knockout. However, if we pre-treat this knockout cell with the LTB4, now we can see there's no any difference. So that suggests TA4H generated at the LTB4, which can, because of the defect. So therefore, there's this phagocytosis defect. Now, if we have a LTB4 adding to the culture, now they restore this response. So this suggests that as long as you have a LTB4, then this phagocytosis effect is okay. So that tells us maybe this pathway here it played an important role. So because the LTB4's receptor is the BLT1, so then we would decide to look at the BLT1. So what we found actually is that the difference expression pattern in the BLT1 is majorly in the neutrophil. If you look at the other cell like BMDM, the peritoneal macrophage, and then other, there's no huge difference. The only huge difference was found in the neutrophil. So that expressed the highest this BLT1. So because that, then we decided to overexpress the BLT1 in raw cell, which is a macrophage-like cell. So as you can see, with overexpressed BLT1 in raw cell, now we can see macrophage, at least this macrophage-like cell, now with or without this the BLT1, that will significantly enhance the phagocytosis activity. Okay, so this tells us that the BLT1 maybe played an important role. So to further provide a genetic evidence for BLT1's effect, we generate the BLT1 knockout mice. So that now you can see with the BLT1 knockout and then challenge with the siabacan, now what we can see is BLT1 knockout, there's a significant defect for antifungal in a immune response because these knockout mice are highly susceptible for the fungal infection. Okay, so just like a LTA4H knockout. So as you can see, this knockout that this kidney has a lot of fungi load. Okay. So now, if we look at the phagocytosis effect, now you can see very similarly. And then with this knockout, now you can see this deficiency of these fungi phagocytosis. But different from the LTA4H knockout, now if you use the LTB4 pre-treat that, there's still cannot rescue this defect. So this tells us somehow this pathway is important role, is BLT1 played an important role in the antifungal immunity. So to summarize this part is the LTA4H deficient neutrophil is with the multi-functional defect on swarming and ROS release and the phagocytosis and killing. And most striking difference is in the phagocytosis. And the LTA4H affect the phagocytosis and ERK activation. So that is only in the neutrophil, but not in the macrophage. So the most likely is due to the difference for the expression of the receptor BLT1. And when we overexpress the BLT1 and in raw cell, that can bypass these can enhance this phagocytosis. And then if we use use LTB4 and can boost this this ERK activation and then phagocytosis with siabacan infection. So now next question is whether BLT1, the receptor, can recognize this siabacan, because we know this cytidectin is a fungi, can recognize fungi's surface component. So we wonder whether BLT1 can do that. And then, but when we always BLT1 in NIH3 cell, so what we can see that, and then challenge with the siabacan, there's no any difference with the challenge. So, but if we use the LTB4, that can activate the ERK in this overexpressed BLT1. 3T3 cell. So that suggests that so BRT1 cannot directly recognize this pathogen. Okay, so but as we know, these neutrophil and then all these C-type-lactin receptor expressed in the neutrophil and the macrophage, they are very similar. Okay, so now the question is, how does BRT1 influence the intrinsic response to the C. albicans or other fungi in the neutrophil? So that's still a question we haven't been able to address. But our preliminary data suggests that if we do the BRT1 overexpressed and then Dactin 1 to 3 overexpressed in the 293 T cell, so we can see what this Dactin 1 can that Dactin family member can associate with BRT1 in the overexpressed system. So so these data suggest that overexpressed with BRT1 in 3T3 cell cannot facilitate cell to activate by C. albicans, suggesting that BRT1 cannot directly recognize the pathogen. And the C-type-lactin receptor were expressed in the very similar level in the neutrophil and the macrophage, but they could not induce the complete activation of the this signaling pathway without BRT1. That's in neutrophil. So in the neutrophil, you have to have this BRT1 to sensing the fungi. So that's something we were thinking whether that maybe this C-type-lactin maybe can talk to the BRT1, because we can see somewhat when they overexpress system, they can co-immunoprecipitate. So that's something we're still working on. So that to give a final model, and then what we see is that, and this LTA4H, which is majorly produced by the macrophage and then monocyte or the neutrophil. So this LTA4H can generate the LTB4, leukotriene B4. This will activate the BRT1. So somehow this one will play the important role in the for the phagocytosis effect of the neutrophil. However, the surprise thing is that with the neutrophil with the Dactin 1, Dactin 2, or all these C-type-lactin is there. However, without the BRT1, now they cannot activate this pathway. So something must be in that it's a neutrophil specific. So one thing we were thinking, maybe somehow this downstream of Dactin pathway in the neutrophil, it's different from other things like a macrophage. So we will find that actually preliminary study is in the SYK, phospho-SYK in the neutrophil, and then we can, if we look at the SYK activation, actually very weak. But in the macrophage here, macrophage there's a very strong. I don't know whether you can see that these somehow blocked by these bar. So anyway, there's a macrophage, and then we can see SYK activation. But in the neutrophil, SYK activation is very weak with the fungi challenging. So that suggests somehow in the neutrophil, there's a BRT1 maybe crosstalk with the C-type-lactin receptor. Just like, there's a G protein coupled receptor may crosstalk with the C-type-lactin receptor. And like there's a previous has an example like a receptor tyrosine kinase pathway can cross to G protein coupled receptor, like they can do the trans activation effect. So we were thinking probably this C-type-lactin receptor may talk to this BRT1, which is a G protein coupled receptor that will bring in the because the BRT1 cannot directly recognize the fungi component. So then maybe the C-type-lactin receptor can recognize the fungi, but in the neutrophil, they require the BRT1 to activate downstream signaling, so therefore enhance this phagocytosis, etc. So that's our final model. And then we were still working on this because this work and still quite preliminary. And then we hope that we can during the next few months, and then we will be able to address all of these questions in the pathway. So this work is mainly done by my postdoc Xin Yan, and then my graduate student Xiong Sihan. And this so that's actually is the only 2 person in my lab working on a fungi infection. And then most of the people in my lab is this day working on other project like inflammation and also anti-cancer immune response. So this is a beautiful campus in Tsinghua. And then I hope anyone, if you were interested to come to China to visit, and then please let me know, and then contact me. I will be happy to host anyone in Beijing. Okay, thank you very much.
Thank you very much, and a really interesting story, and I'm sure there will be lots of questions coming. So just to remind everyone, as Sarah has already said, and I've said earlier, please put your questions into the Q&A function that you'll see at the bottom of your screen. And at this stage, it would be quite useful if you could just indicate who your questions are directed to. And Mary, you can come back. There you are. Great.
Okay, sorry, just saying you can stop sharing your screen, and people put them in the Q&A, not the chat. The chat's not being monitored. I see already there's one question in the chat.
So
please put that.
I just do I just stop my sharing?
Yeah.
Yes, please. Yes, please.
Okay. Okay.
Okay, I'll start off the questions, Mary. I've got a couple for you, which I know you've answered in writing, but nevertheless, the audience won't have seen those. So I'll just ask those questions again directly. So there's a question asking about the virulent versus avirulent candida and why this is not inducing the GR1 myeloid suppressive response, and whether they are induced by both of these organisms or there's a mechanism that is suppressing them.
Yeah, so this this whole differential ability to protect was pretty shocking. But one of the first things that we noticed when we started looking in the bone marrow is that my lab manager, Elizabeth, when she was plating out bones, she noticed that when we had vaccinated with Candida albicans, the bones and the bone marrow appeared, the bones were more fragile and the bone marrow appeared bloodier. And so we knew that there was something dramatically different between Candida albicans and Candida doubleriensis in terms of what was happening in the bone marrow. And we just suspected it was all related to the level of damage that was being induced by the 2 different strains. Clearly, when you take out EFG1 and CPH1 from albicans, it's now able to induce protection. So it's definitely a virulence factor related phenomenon. We think that beyond causing more damage, that perhaps there's also cell death being induced. So we're looking at markers of ferroptosis in the bone marrow and whether there's differences between the different strains in those patterns of cell death within the bone marrow. So probably multiple things going on as to why albicans is unable to induce this nice protection.
So can I just push you a little bit on that? So then are you actually seeing this GR1 MDSCs being induced, even though you're getting this big messed up bone marrow? Do you actually see those in
right, to some level, but not to the degree that we see it with doubleriensis. So I think that they are actually causing death of some of these MDSC precursors in the bone marrow. So it's not just a quality issue, it's a quantity issue. I think that they could induce them to some level, but since they're killing off, part of what, the cells in the bone marrow, that's why it's not, it doesn't work as well with albicans. So, if we can relegate it to completely avirulent, which is our most weak strain is that double mutant, then it does function. So.
Okay, thanks, Mary. Just before I hand over to Sarah, so somebody's got their hand up, I noticed. We can't take live questions. Please, can you put your question into the chat if you want it to be addressed?
No, the Q&A, not the chat.
I'm sorry. Yes, I say it's Q&A every time, and I've just said chat. Into the Q&A, please, if you wouldn't mind, and we'll take it from there. Over to you, Sarah.
Yes, thanks. So a lot of questions, including for myself, about this concept of being a co-receptor. What about the FC receptors? Is there any interactions with that?
Yeah, so we haven't go to that part. So that's obviously that's a question. So somehow they probably need to have a downstream component to talk to that. So, but BLT1 is a G protein couple receptor, so that can hook up a lot of the different signaling component. But the C-type pathway is a talk to FC receptor. So those pathway. So we try to, right now we haven't, so I just showed the SYK. So one thing is we need to work upstream of SYK, for example, like FC gamma or these even the SAC family kinase or those things. So that's something we try to work step by step.
Right. But related to that, and maybe you said it, but does the knockout of the BLTs affect overall expression of any of the ductin receptors? Are there trafficking or anything?
So in the wild type and the knockout and then situation with or without stimulation, there's not dramatic. There's no significant difference in terms of these C-type receptor. So they are very.
It's not a fundamental cell biology problem.
Yeah. So.
Great, thanks. Over to you, Gordon.
Murray, so I have a question here about the depletion of the GR positive cells during the second challenge, and does that lead to increases in pro-inflammatory cytokines following vaccination if you do a second round of depletion or you deplete at a later stage? You're on mute, Murray.
I would suspect that it would correlate with reduced cytokine levels. It's hard to tease out, that the problem with MDSCs in the murine model is that the markers for inflammatory neutrophils and GMDSCs are identical. So there's no way that we can selectively remove just the MDSCs and leave the PMNs intact. So that's been, a hurdle that we've been trying to get around. Everyone in the community is trying to get around this. It's kind of the holy grail of MDSC research. So, there's the yin and yang. So if you deplete all of them, you're also depleting inflammatory antimicrobial neutrophils as well. But in our case, getting rid of them abrogates protection and abrogates the ability of IL-10 to function to suppress the immune response. So it's a really tricky two-sided equation when you're talking about these, Ly6G positive cells. So that's kind of why we went after the suppressor phenotype with IL-10.
Okay, thank you. Sarah?
So Singh, are there other immune baseline phenotypes in either of these knockouts?
Okay, so the mice looks perfect fine. So although this is so important, but they look perfect fine. So that to answer this one of the questions.
Okay, well, it's nice for these kinds of studies. All right, and since that was a short question, there's a question about sort of broadly about inflammatory resolution, thinking of this as a negative regulator, and specifically whether you looked at the aminopeptidase activity of LTH4H, which degrades, as you know, degrades the some of the neutrophil chemoattractants and promotes resolution. So if you look at long-term, is there more immunopathology, I guess?
Yeah, I think this is a very important question. So right now, we haven't go to that far yet. So we still work on very this phenotypic work. And then we really need to go to the detail to look at this enzymatic activity, whether that's has some rule there or not. So we haven't go to that far yet. So
the vaginal model might be an interesting place to do that.
Yeah. And also, so the other question.
We'll swap over to Gordon.
Okay, just
Right, Mary, there was a question that you'd also had answered, but also just for the general audience, it was around adoptive transfer of the CD4 positive cells, where these cells, if they're trained ex vivo with fungal ligand, can they confer protection?
Right. So we had originally had a whole section of the AIM, ex vivo, trying to train bone marrow cells with albicans or any of the other Candida species, and then eventually, do a lot of the cytokine analysis and marker and maturation analysis, and then hopefully get those transferred in to the animals. And in the meantime, we just started isolating, different tissues and doing it that way. This was way prior to when we actually showed that beta glucan as a ligand could actually induce strong protection protection. So we were only at that point working with intact microbes, or we were separating them, you know, in a dish. So we, you know, we couldn't separate the microbes, since we knew it had to be live to induce protection from the cell to adaptively transfer those precursors. It's something that we could revisit at this point.
Okay, Sarah.
So there's a question about the intracellular calcium response.
Yeah. So this is something we haven't worked on. As we know that this G protein coupled receptor can induce the PLC activity, and then also increase the calcium function. And we haven't got to see whether that there's a crosstalk between the C-type-lactin receptor and then these induce the calcium function. So we haven't worked on that yet. So that's something certainly we need to look at that. Okay.
So Maria, I have a question for you, my own question, which is related to this IL-10 phenotype. So your only conclusive evidence of that was from the IL-10 knockout mouse, which are pretty messed up, as you know.
Yeah.
Do you think that's the only mechanism that's operating here? Because if that's true, then theoretically you just add IL-10 to control the sepsis by adding in IL-10 to control it, right?
Right. We think it's a major mechanism. There could be other anti-inflammatory cytokines involved, and we haven't done knockouts in any of the other pathways that are known to be used by MDSCs, at least in other models. So in a sepsis model, it's, you know, from things that I've read, it's predominantly IL-10 mediated suppression, and that's kind of been a linchpin. On the other side of the coin, what elicits this dramatic inflammation, we know that part of it, I didn't present that, that is PGE2 specifically. So if we, you know, hit that, you know, so we haven't gone back to try to hit that, you know, in this model as well, in terms of the protection model. So right now, this was the very first, you know, mediator, and thankfully we did get a phenotype, but we're probably going to have to back that up with IL-10 antibody mediated depletion as well, because those 10 knockouts are, you know, from birth, not in good shape.
So I mean, you know, you talked about the sort of translational therapeutic angle at the end of your talk.
Mm-hmm.
And wouldn't IL-10 administration be sufficient to, I mean, why bothering with all this, trying to get beta-glucan or fungi?
Right. I think,
why not just treat them with IL-10?
yeah, I think that's an interesting idea. I know that, you know, even with the early stages of the COVID pandemic, when people realized that the markers that were being upregulated and people that were dying were very much markers of sepsis. So IL-6, you know, that sort of thing. And the thought was this, like, let's hit the inflammatory response with anti-IL-6 in humans. And it was very much hit or miss. So I think there's a very localized effect of these cytokines, so that if you just bolus administer, you know, a cytokine or an antibody, it might not get to the location where you need it at the time you need it. So in our cases, trying to treat some of these mice prophylactically with an NSAID, which is what we had been doing originally, you have to give it within a few hours of the inoculation. That's not feasible in patients. So our concept was to use, you know, a live attenuated vaccine that could induce these MDSCs, whether it be, you know, BCG, you know, is being looked at for the same reasons in Europe, and it was part of a clinical trial that was a very large clinical trial across multiple countries to use BCG as a prophylactic to prevent the inflammation associated with COVID. And so very similar concept was involved there. So if you vaccinate people with it, then they already have the cells in place that can come in where they need to go and block, you know, and control inflammation. I have my doubts that administering just one cytokine will actually do what a cell could do. Same thing with, you know, vaccine is the ultimate way to prevent, you know, an infection. There's multiple ways where vaccines work. In our situation, we're trying to block inflammation. So we think that something like this conceptually might last about a year. Same thing with probiotic that you give orally, you have to take it every day or every week to actually have its effect. As probiotic that would induce these shorter lived MDSCs, you know, it's not lifelong immunity, but could last perhaps up to a year in humans. So those are kind of the concepts that have been thrown around in the BCG community as well.
Okay. Thank you. I'll come back to that in a moment after Sarah's asked her next question.
So I have a philosophical question, given especially that, particularly with Gordon and Singh here, but it's always been amazing to me that there are such parallels between the T cell receptor signaling pathway and dectins, right? But, you know, you look at the TLRs, they're classic or nod-like receptors, and they're really different. Why? I mean, I realize obviously, we can't really answer that why, but I'm sure this has occurred to you. Why do you think these responses to fungi are so analogous to T cell receptors and presumably evolutionarily predate them?
Right.
Or put another way, why did sort of our adaptive immune system, T cells and B cells, co-opt a fungal sensing signaling mechanism as opposed to anything else? Because we think of it naively as all just converging to get, to NF-kappa B and NFAT. And there are other ways to do that, right? So why, is there something that is special about that utterly complicated way of activating signaling that is better tuned for those types of, of responses?
So I can't speak for the B cell because I think B cell has both, but T cells have ZAP70 and myeloid cells have SIC, and their structural constraints of their 2SH2 domains are very different. And I think that is the evolutionarily difference. I don't know whether Zin can comment, but the centralized signaling pathway that's used by all C-type lectins that induces actually very different responses. I think it's a lot more simple than a lot more complicated than the simple idea we have in terms of the way it works for sure. And work like we're hearing from Zin and from many others are starting to get some insights into that and some of the other pathways that are involved, but it's oversimplified as always. But in terms of your original question, I think it's just an evolution of a pathway that's been useful for activating cells in response to danger.
Yeah. So I think that Gordon is right. So it's for the innate cell, which is using this CLR and those things to talk to outside this different variety of the component. So that way you can recognize it by C-type lectin. So that way you can have it all different. But in terms of signaling, so there's certainly some similar component versus TCR or BCR. They either TCR will work on downstream of this is ZAP70, and then those like similar to BCR SIC. So that's a pathway using the adaptive immunity. This is evolutionary after this with the T or B cell and with a specific, motif that can recognize a much higher specificity like a TCR, like a BCR. So that way, but downstream they want to use a very simplified pathway, common pathway to trigger the these JNK, ERK, and kappa B. So they want to utilize this machine, and then it's using quite simplified machine to make the cell growth. Basically, it's growth signaling pathway. So that's, and then also anti, pro survival pathway. So these are all either growth or provide a survival pathway. So that's why they are very similar. After you want to mobilize your innate immune response, you mobilize your T cell, B cell. So that's what happened. So that's my simple view of this signaling receptor. Yeah. Yeah.
Oh, you're probably the only person on the planet who would say T cell receptor signaling is simple.
Oh yeah. So we've been as you know, I've been studying the TCR and for more than 20 years. Then CLR like a Gordon, and then I've been working on that for about 10 years. So both this pathway, and then we keep working on that. Now this day we use the artificial TCR, this is so CAR T, and then we call also STAR T, and those modulated TCR. So we use it as a tool to target different things right now. Okay. Yeah.
Are you trying to develop CAR Ts for fungal infections?
It's more difficult. It's too costly. So I don't suggest to use that. Yeah.
All right, should we have one more question from me again? Sorry, Murray, and then bring the session to a close. So I wanted to ask you a bit of a wacky question, a philosophical question in a way. So there's a lot of literature going back actually to the early 60s about the impacts of beta glucans on hematopoiesis. Okay. And there's been a company that's set up, as you probably are aware, that are using beta glucan to treat. That's all related to this innate immune memory. But I was wondering about the food, because there's also evidence that all of the murine child that we have, there's a lot of beta glucans that translocate across into the bloodstream. And I was wondering whether you had thought about that in terms of your models, right? And whether you had looked at that on mice without beta glucans in their diet.
Right, right. So we haven't looked at that, and beta glucans are, a generic broad group of carbohydrates that can be found in not just fungi, but also in plants. And so like my my lotion that's on my desk here says comes with beta glucans. So they're almost in like all different types of products, and they, have been used homeopathically, as you know, for a long time. And now we're a little bit more educated in what they can do in the systemic milieu, which is kind of surprising. So a lot of, there's a lot of beta glucan oral delivery, and it's supposed to, tone down inflammation, and, who knows how many of those have been founded on, controlled trial, from trial data. But thinking about how, it could actually bypass the GI tract and get into the systemic environment. And maybe that's part of why oral delivery beta glucans actually does what it can do in some people. I mean, it seems to be more anecdotal than not, but it will be interesting moving forward. So the beta glucan that we've been using in our studies, we just purchase it to make sure that it's, the same lot every single time, so that there's not a huge amount of variation. So it's from Saccharomyces cerevisiae. We're not specifically using glucan from Candida dubliniensis, but it seems to be, it does need to be fungal derived. What I didn't have time to go into today was my student actually had modified that beta glucan that she ordered from Sigma. So she used an alkalinization procedure in which we were using that to basically label the beta glucan and follow it into the bone marrow and see what cells were interacting with it. And in that process, she decided to challenge those mice that she had administered that alkalinized glucan, and it worked really well. The regular untreated beta glucan that we purchased from Sigma did not work. And similarly, we have to use depleted zymosan, which is alkalinized as well, to remove TLR signaling, and that's the one that works. So in both cases, we treated the glucans to try to basically end up removing TLR signaling, and we've been showing that with reporter cells. So if you hit TLR pathway plus CLR pathway, it does not work the way it should if you just eliminate that and only go through the CLR pathway. So
that's very interesting. So these
we got
pathways,
lucky.
they definitely modulate each other.
Yes.
That is a very interesting observation.
Yeah, and you can reverse tolerance induced by glucan administration by hitting with LPS afterward. So there's some old studies looking at that as well. Also, we tried to do an LPS endotoxemia model and see if we could induce protection against that, and it does not work. So there's something very different between an endotoxin type sepsis acute mortality model and other types of sepsis that we can induce either through infection, or we've used zymosan to induce that too. So some things that definitely need to be worked out between those two divergent, recognition pathways.
Yeah, something really interesting. Well, thank you, Mary and Zen. Thank you both very much for coming and speaking.
Thank you.
It was a really enjoyable
very
session.
much. Yeah.
And thank you to the audience for being part of the discussion and for attending. And we'll see you all next month.
And
Bye,
to the
everyone.
crew behind the scenes at Exeter who make this happen.
Thank you.
Thank you, everyone.
Bye-bye.
Okay. Bye-bye. Bye, everyone.
Bye.
Okay.
Good night.
Okay.
Saying it's late for you.
Okay. Good night. Good night.